Viruses are tiny parasites that invade host cells and co-opt them in order to reproduce. How do they do this? Imagine the cells that line your blood vessels or your intestines. These cells are called endothelial cells. Here is an endothelial cell just hanging out, doing its thing, making ATP, making proteins, and to this cell, everything is just grand. But unbeknownst to this cell, lurking in the shadows, is the villain of our story, Ebola virus. Ebola is really tiny. If you were the size of the Ebola virus, our endothelial cell would be the size of the Empire State Building. Nearly 8 million of you could fit inside the endothelial cell. Now, let's take a closer look at the Ebola virus. Each Ebola virus is called a particle. Like all living things, Ebola has an instruction manual that tells it how to reproduce. This instruction manual is the virus genome. For Ebola, its genome is made of RNA. The genome is protected by a structure made of proteins called the capsid. Stuck onto the end of the genome capsid structure is a protein Ebola uses to make mRNA and genome copies. This protein is called an RNA polymerase. Different RNA viruses have their own RNA polymerase. Ebola's RNA polymerase only works on its own RNA genome. Finally, Ebola has an outer layer made of proteins and lipids called the envelope. Not all viruses have envelopes. So, going back to our story, the endothelial cell is going about its business, and everything seems normal. But in order to reproduce, our villain, the Ebola virus, needs to get inside the endothelial cell. Luckily, it carries a key to unlock the endothelial cell membrane. This key is called the glycoprotein. Ebola's glycoprotein fits into particular host proteins on the surface of the endothelial cell. We'll call them locks. Once the Ebola glycoprotein engages the lock, this triggers the endothelial cell to start swallowing the Ebola virus whole. This process of swallowing is called endocytosis. And when it is finished, Ebola is sitting inside the endothelial cell, surrounded by cell membrane. Endocytosis is a normal part of everyday cell activity. Cells use endocytosis to take up nutrients that can't go through the cell membrane on their own. Iron is an example of one such nutrient. To make a whole new virus particle, Ebola needs to do two things. First, it needs to reproduce its instruction manual, or the genome, and second, it needs to reproduce its structure, like its protective capsid and envelope. Like many viruses, Ebola is lean and mean. Since the virus is so small, it doesn't have room to carry around the machinery it would need to reproduce itself. Instead, Ebola hijacks the machinery that the host cell uses to make proteins and membranes. Once Ebola has hijacked the host cell, it turns it into a virus factory. These virus factories work as hard as they can to churn out copies of the virus. In our story, an Ebola virus is now floating around inside the endothelial cell, protected by membrane, or a vesicle, so the endothelial cell doesn't really know that Ebola is even there. Next, the Ebola virus injects its instruction manual, the RNA genome, out of the vesicle into the cell's cytoplasm. The RNA polymerase that Ebola carries with its genome starts to transcribe the genome, making messenger RNA. Now, the endothelial cell starts to encounter problems. Its cellular machinery has stopped making endothelial proteins and has turned into a virus factory, churning out only viral proteins, and these proteins are building up at an alarming rate. Soon, the cell's surface is studded with clusters of the Ebola glycoprotein. This starts to interrupt the endothelial cell's connections with its neighbors. Once a quota of capsid proteins has been made, the Ebola RNA polymerase stops making mRNA and starts to make copies of the whole genome. Next, The viral proteins and genome copies begin to assemble on the inside of the endothelial cell underneath the glycoprotein clusters. When a complete Ebola virus particle has assembled, the endothelial cell burps off the Ebola particle in an opposite way to how it swallowed the virus at first. Notice that the Ebola particle has taken some of the cell membrane with it. Soon, the cell is burping out hundreds and thousands of viral particles. And at this point, the endothelial cell's life is over. Our villain, Ebola virus, has won. With one cell turning out so many copies of a single virus that go on to infect other cells, and then they churn out hundreds of new viruses, you can see how this would quickly get out of hand and cause a person to get sick. Luckily, our immune systems help keep viral infections in check. But that's a story for another time. So let's review. What did Ebola have to do in order to hijack the endothelial cell and turn it into a virus factory? Step 1. Ebola attached to the cell surface using its glycoprotein key. Different viruses have different keys that allow them to attach to different locks on different types of cells. Ebola has a key that allows it to enter endothelial cells that line blood vessels. This virus causes symptoms of bleeding. Alternatively, the influenza virus has a key that unlocks cells located in the respiratory system. This virus causes respiratory symptoms. Norovirus attaches to the cells in the intestine. What kind of symptoms do you think it will cause? Step 2. Ebola entered the cell through the normal process of endocytosis. Once it was inside the cytoplasm, Ebola injected its genome and its RNA polymerase into the endothelial cell cytoplasm. Not all viruses use endocytosis to get inside of cells, but it is the most common method. Step 3. Ebola hijacked the cell's own protein-making machinery, stopping the cell from making its own proteins and forcing it to make only viral proteins. How a virus takes over the host cell is highly varied. Each virus has a different strategy, and each strategy is clever and elegant. Step 4. Ebola exited the cell. It did this by putting envelope proteins into the cell membrane and then clustering virus components underneath the membrane. Then Ebola burped, or more elegantly, butted off virus particles, taking some of the cell membrane with it. This process of butting off is what gives Ebola its envelope. Viruses that are enveloped, like Ebola, don't always kill cells immediately. The cell can often survive a couple of burps. But not all viruses are enveloped like Ebola. Viruses that aren't enveloped burst through the host cell, leading to cell lysis, a very dramatic and catastrophic exit that kills the host cell immediately. Here's some food for thought. Viruses don't need nutrients, and they can't reproduce on their own. So are viruses alive? How then would you define the word life?